The kind of close-up talk of the symposium is by Professor MacKinnon from the Rockefeller University. So Professor MacKinnon has been one of the real pioneers and leaders in understanding the structure and function of ion transport membrane proteins and membrane proteins in general, and was awarded the Nobel Prize for this work in chemistry in 2003. And I think it's great honor and privilege to have Professor MacKinnon kind of wrapping up this meeting, honoring and celebrating the 50th anniversary of the PDB. I should say also Professor Rockefeller University and Howard Hughes Medical Institute. And so the title of the presentation is The Incredible Diversity of Potassium Channels.
Okay, if you're hearing this lecture as a pre-recorded version, I apologize. I knew that I would be in a place where I'd probably only have cellular connection during the meeting. And so as a safety measure, I pre-recorded this. But in this case, if you're hearing the pre-recorded version, I certainly hope I will be here for the Q&A. The subject is diversity of potassium channels. And unlike my usual detailed mechanistic talk on a single thing, I've decided to give an overview where I don't go into too much detail, but I think it's very appropriate for this occasion because it's been fun to think back on our efforts on potassium channel structures. My lab has been using structural methods for about 25 years, and we've seen a lot of change over that period. In the beginning, after you could figure out how to purify and isolate a protein to try to crystallize it, it was a many-year process of growing good enough crystals, synchrotron data collection to improve the crystals, phase determination, model building, refinement, a very slow process. And then, as you've all seen, with the advances in cryo-electron microscopy, this process has become much, much faster. And most recently, with a good progress in structure prediction from amino acid sequence, we've seen even further change. So there's been a lot of change, and I want to give a little bit of this historical perspective and also tell you where I think, at least I am going with in our structure work. So onto the subject, diversity of potassium channels. 1998, we got to see the first structure of a potassium channel, and in fact, the first structure of any selective ion channel. And this was the KCSA potassium channel from Streptomyces lividans, shown here, a small tetrameric protein whose essential feature is that it contained a selectivity filter that catalyzes the fundamental process in potassium channels, which is the selective conduction of potassium ions. This selectivity filter is made by a highly conserved sequence that makes an unusual configuration of the main chain, pointing carbonyls towards the center to make 4 ion binding sites where potassium fit in a dehydrated state, and potassium diffuses across the membrane by hopping from one site to the next in this selectivity filter. Now, fast forward 20 years, and this is showing a dendrogram of the family of eukaryotic potassium channels. And you can see that the potassium channels, in terms of their structure, are highly diverse. But what they all share is that same pore with the highly conserved selectivity filter. And in fact, if you look in any of these and superimpose the selectivity filter, you see that the oxygen atoms from the selectivity filter superimpose almost perfectly at an atomic level. So in evolution, that part of the potassium channel, along with the surrounding important features of the surrounding immediate pore, were highly conserved. But then attached to that pore are various domains that allow the potassium channels to sense different signals in their environment. And the reason for this diversity, of course, is that potassium channels fill a myriad roles in physiology. A little historical perspective on this. These four structures, there are more than this, and these are representative ones from each clade of the potassium channel family. But these four shown with green text were determined by X-ray crystallography. And the first thing to notice is that the first of these was solved in 2005. So KCSA was 1998. So the 7-year gap between KCSA and a few other following bacterial potassium channels, the 7-year gap reflected the time it took to figure out how to express and purify recombinant eukaryotic membrane proteins. And notice also that these four structures came over a period of about 8 years. So it was slow going. And that time period reflects the difficulty in getting good quality crystals of these eukaryotic membrane proteins. Then, of course, after 2014, everything changed. So the structures shown with blue text are solved by cryo-electron microscopy, single particle cryo-EM. And notice that these many structures came over a relatively short period of time, reflecting the fact that you don't have to figure out how to get good crystals. And there are other advantages, of course, to not having to crystallize your protein in terms of allowing the protein to undergo conformational changes. To this diversity of structure and connected to the diversity of function, I show here a little overview in red text of some of the roles potassium channels play. The clade up here, these are calcium activated potassium channels, and I'll show you a little bit more about those in a minute. These are called inward rectifier potassium channels. They are regulated by signaling lipids, TYP2, and some of them by G protein subunits. So they come under the control of G protein coupled receptor stimulation. TRAC is an unusual looking potassium channel, and yet its selectivity filter is highly conserved in structure. It exists in the node of Ranvier and plays thus a role in the electrical activity of the active unit of myelinated nerves. These are various voltage-dependent potassium channels. Actually, HCN has a modified selectivity filter and lets sodium as well as potassium through. But they all play a role in different cells and also in different parts of cells. So KV1, for example, is involved in nerve electrical impulse conduction in axons and cell bodies, whereas a channel like KCNQ plays a role in conduction in the heart and the nervous system. EAG, a calcium-inhibited potassium channel, probably plays a role in very specialized parts of neurons related to synaptic vesicle release. And so you get the idea that these have evolved to fill many roles. Now, let me show you a few examples just to get a sense of how these work as little machines, and again, not in too much detail. This is a calcium-activated potassium channel called SLO1. It plays a role in skeletal muscle repolarization and also in auditory tuning in the cochlea. This channel is suspended in the membrane. So here, the pore with some other actually units that I'll come to in just a few minutes, suspended in the membrane with a large cytoplasmic domain pointed to the cytoplasm. This channel opens when intracellular calcium increases, and I'll show you a morph movie between the closed calcium-free conformation and the calcium-bound conformation here. And so you get a sense of the 3D structure. Calcium binds, And this unit in the cytoplasm, we call a gating ring, binds calcium, undergoes a large conformational change, and that conformational change does work on the pore here to open these helices up and thus open the gate to let potassium conduct. Now, let's go to a different calcium activated channel. This is called SK. It's important for spike repolarization, spike frequency modulation in some central neurons. And here again, it's a potassium channel. It's activated by calcium. And I show you this one because it gives an example of how nature was sort of, in a sense, what comes out looking very creative in the way these channels evolved. Calcium activated, but the machinery to sense calcium in this case is completely different than the slow one channel we just saw. In this case, the purple domains are actually calmodulin, 4 of them, 1 to each subunit, bound at the cytoplasmic membrane layer. And these, so essentially this channel has recruited calmodulin to bind as its calcium sensing machinery and to open the channel. And again, I'll show you both between the calcium free states and the calcium bound open state. So when we first look at this through cryo-EM, we can see multiple structures of the calcium free state. The end lobe of the calmodulin is free to move. The C lobe is bound tightly to the channel. But when calcium binds to the end lobe, you'll see that it undergoes a conformational change. And so the end lobe opens up. And when it opens up, it exposes residues that then bind to the helices that are closer to the porous gate. And when that binds, it's associated with an expansion of the pore. So you can see that the cytoplasmic level of the channel expands, opening the pore. And if we look down the fourfold axis of the channel, you'll see how the pore opens when the calcium-bound calmodulin binds. So this is a totally independent way that nature came up with a calcium-dependent potassium channel. Many of the potassium channels, and in fact other ion channels like sodium and calcium channels, are what we call voltage dependent. And in this case, I show you a cartoon because unless you're in this business of membrane excitability, the concept of what a voltage sensor is and what voltage dependence is pretty unfamiliar. But it's a very simple concept. The membrane protein, the channel, is of course suspended in the membrane, and the cell membrane is a good electrical capacitor, meaning when it is charged and the source of charging of the ion gradients moving through channels, there's a voltage difference across the membrane, creating an electric field inside it. And certain ion channels that we call voltage dependent channels evolved a way of sensing that electric field. And essentially it's a domain depicted here with charged amino acids in it that get driven from one side, either outward or inward. And that movement of the voltage sensor is coupled to opening and closing of the pore. So in a sense, what I'm describing here is nature's field effect transistor. And in the analogy to a field effect transistor, the membrane voltage is the base voltage, and the conductance is the pore that is gated, opened, and closed. And some of these are exquisitely sensitive. So here I show you a KV channel, a voltage-dependent potassium channel current, as a function of membrane voltage. And you can see that that current that is almost off at minus 100 millivolts is almost completely open by, say, minus 75, minus 70 millivolts. So it's a very steep switch-like turn on. And this is important for what these channels do. So how did nature come up with this voltage sensor? What does it look like? We got to see those first in voltage-dependent potassium channels. And here is one such voltage sensor removed from the pore. It consists of 4 main helices, and one of these helices, called the S4, because it's the 4th helix in the voltage sensor, is hydrophobic, but it also has basic residues, typically arginine, with occasionally a polar substitution at every 3rd position. And a hint as to why this is every 3rd position is part of this helix adopts a 310 configuration as it passes through the middle. So the concept here is, and it's been demonstrated in a variety of structures in a number of labs, that this helix moves, and the movement of the helix, driven by the transmembrane voltage difference, works on the pore to open and close it. Nature has been pretty creative in the way it adapts this voltage sensor. So the voltage sensor is always, they're always a similar structure. It's a highly conserved unit, but it's adapts to the pore in different ways. And we found 2 important, fundamentally different ways. It attaches by what we call direct and domain swapped. And in the direct attachment, the voltage sensor is associated with the pore from the same polypeptide chain, whereas in the domain swapped, the voltage sensor is associated with the pore of an adjacent same polypeptide chain, but in an adjacent position. So these are the voltage sensors are in a sense domain swapped around the ring of subunits, and these allow the channel, the voltage sensors, to engage the pore in a different way, and it's a topic that we and others still study. I want to, as the last example, I show you the G protein gated potassium channel. And you'll see I showed this last because it gives me reason to explain where I think we are going in these sorts of membrane protein structure studies. So here in the G protein gated channel, the transmembrane pore crosses the membrane. We recognize the pore that we first saw in KCSA with its selectivity filter. In the cytoplasm, there's a large domain, and that domain can bind beta gamma subunits, G protein subunits. You also see sodium and PIP2, the other ligands that activate this channel. So we know the structure in the presence of all of its activating ligands. We know the structure in the absence of the activating ligands. We know the mechanical conformational changes that switch this from a closed to an open channel. So in terms of a single molecule working in the membrane, we know a lot about this. I mean, it's a very well-described system. And if we look at what this channel does, show you a little physiology here. On the top is a recording from a sinoatrial node pacemaker cell, isolated from mouse heart. Single cell in a dish with a recording electrode on it, recording membrane voltage on the y-axis and time on the x-axis. And what you can see is this spontaneously depolarizes, and this is the kind of cell that keeps your heart going. Isoproterenol is supplied and it speeds up. The isoproterenol is removed, and then acetylcholine is applied, and the membrane hyperpolarizes, and the spike polarization, the frequency goes to 0. In an intact organism like us, both of these stimuli, the isoproterenol and noradrenaline and acetylcholine, are always acting at some level so that the heart slows down or speeds up, but hopefully doesn't stop. Acetylcholine here is working through the G protein gated potassium channel. And in fact, if you take the same cell and you now, instead of recording the membrane voltage, you switch to what's called voltage clamp mode, and you apply acetylcholine and measure the current, what you see is acetylcholine application associated with a large potassium current, outward potassium current. And this potassium current is what hyperpolarizes the membrane, slows the heart. Now, the way this works is that acetylcholine is binding to the M2 muscarinic G protein coupled receptor. That catalyzes guanine nucleotide exchange in the G protein heterotrimer releasing the alpha and beta gamma subunits, which diffuse in 2 dimensions along the surface of the membrane. The G beta gamma subunit finds the potassium channel and binds to it and causes it to open. Now we know from the literature and from our own work, for a typical SA node cell, we know the average density of the M2 muscarinic receptor in the cell. We know the turnover rates, so that when you stimulate the receptor, we know how quickly you generate G beta gamma. We know the diffusion coefficients of G beta gamma across the membrane, in the membrane, from direct measurements. And we also know the affinity from our own work of G beta gamma to the GERK potassium channel. We know that 4-bind, we know the cooperativity factors for this binding. So we know what would seem to be everything about this pathway. And yet the truth is, if you try to reconstitute the pathway, or if you model this pathway, plugging in all the numbers that we know that are fairly accurately measured, it does not work. And what happens is, given the density of G protein coupled receptors on average across the cell, and the rate at which a stimulated receptor can generate G beta gamma, and the finite lifetime of G beta gamma, that is, it's only free for a second or two before the alpha subunit catalyzes the hydrolysis of GTP, and it again resequesters the G beta gamma. And knowing the affinities and cooperativity factors, you never get enough G protein, G beta gamma to activate GERK. So we seem to know everything at a detailed structural level about this system. And yet, if we model it, it doesn't work. Now, it turns out it can work if you invoke something that we don't know about. And that is if we invoke a specific organization of the G protein coupled receptors. You could call it clustering, but it doesn't have to be. It has to be an arrangement of these receptors on the cell surface that is not random. Now, it's not as simple as, okay, what if you have a G protein gated channel bound to a GPCR in a macromolecular complex? It turns out if you make that macromolecular complex, it still doesn't work. That's not what explains it. And the reason is, given the catalytic rate of turnover, you never get enough G beta gamma on the inside of the membrane, close to a single GPCR to activate GERK, even if GERK is there. And so what this implies is that there's some structural organization in the cell membrane, probably in the few hundred nanometer length scale. And that's the length scale that we don't know very much about. Of course, because of X-ray crystallography and cryo-electron microscopy, we know a lot about macromolecular structures. We know how organized they are. We've seen many of them. At the scale in which we can use a light microscope, we know a lot about the organization of cells, organelles, all the way up to organs. But right in this gap in between, a couple hundred nanometers and the angstrom level, we don't, or say 10 nanometer level, we don't know very much about the organization in cells. And I think the real key to understanding a pathway like this, and probably nearly all biochemical processes, we're going to have to understand structure at this length scale. And so I would say that we have to determine structures of interacting molecules on this length scale. For that reason, I actually think it's an extremely exciting time in structural biology, because the way I would put it, it's the first time that methods are in place so that we can begin to introduce the space dimensions, not just the time dimension, to the biochemical rate equations. So I'm extremely optimistic, actually, and I'm very excited to be around and have energy to work on these problems now, because I think we're going to learn a lot in the next 5 to 10 years. Okay, I just will end. This was a very general talk, so I'm not listing names, but I've worked with many wonderful scientists over these years. And also, I thank HHMI, NIH, and Rockefeller University for supporting the research. Now, hopefully I'm here to take questions. Thanks very much.
Okay. Well, thank you very much, Rod. It's really amazing to see the evolution of the whole story and the level that it comes to, from structure to really to cell biology. So I was actually quite impressed with the efforts to model the GERK potassium channel pathway. And I wonder, when you started to use the word density, and I wonder if it's the case that many ion channels have even simple oligomeric interactions which modulate their function. When we study them even in cryo-EM, we don't have that piece of the story. Can you comment on that? Do you think this is a very general feature, and that really many ion channels are going to be modulated by such oligomerization or clustering?
I do. I think probably most membrane proteins, won't be randomly distributed. In fact, they'll be very not randomly distributed, and it'll be central to understanding how they work. In this case, it's mostly the G protein coupled receptor that is not working in isolation. In order to generate enough G beta gamma inside the membrane, they essentially have to be in a cluster or some organization. So a single one, it's a simple problem of saying, if I generate GPCRs at a given rate at a source, and I know the diffusion coefficient and the lifetime, I can calculate what the profile of concentration is radially away from that GPCR, and it never achieves a high enough concentration. So but then you say, what happens if I bring a little group of them together? Of course, then you get a solution. You essentially get the sum of those things, and that sum starts to build up. And so the way you can get away from this is to say the GPCRs have to be in some sort of grouping, and that's sort of a big question we study now. And I think, as to your question specifically, I think the organization on the cell membrane won't be random, and it's going to be very, very interesting. Of course, this has been a kind of blind spot, because using the techniques to look at organization at the atomic level, X-rays and electrons, we can see things very well. And then, of course, using light microscopy, we can see things very well down to the diffraction limit of light, visible light. But then we have had a gap in between without the great techniques to fill that in. And this is the range, the scale that I'm talking about, up to a few hundred nanometers, that I think is going to have the essence of how many signaling and biochemical pathways in general work. As I said at the end, I really think that, for the history of biochemistry, our kinetic equations only have the time variable, and it never has the space variable, because we haven't had that. But now, for the first time, methods are coming into place to get that space variable. And I think electron tomography is going to be an extremely important method for that. And I think then we can get directly at the question you're asking about distributions on the membrane. And I think it's going to be fascinating.
Yeah, a related question is from Rag Harriman, who has asked, are you referring to lipid rafts when you talk about the 200 nanometer timescale origin?
Yeah, we study them. We're not quite sure. So you can ask obviously we have a belief, and in fact, we have some data that things are not randomly organized and that things do get clustered. And then of course, ultimately, we want to understand how cells do that. And we have a few, we and others have a few hypotheses about how it works. And one of those certainly is lipid rafts. And we study lipid rafts in the lab in giant unilamellar vesicles. and we study them in black lipid membranes that we make use of in the lab. So we try to reconstitute these whole systems, under a circumstance in which we can put fluorophores on the components and look at them move around. And RAFs are very interesting. We're not really sure whether it's one of the possibilities for clustering. We're not sure whether it is really how it works, what cells do, but it is a possibility, and we study it. So I think they're interesting. The membrane is certainly heterogeneous for many reasons. The other obvious mechanism of clustering of organizing membrane proteins is through tethers to cytosolic proteins. And there are certain cases where we know, for example, postsynaptic density proteins are what help organize proteins in the postsynapse. And so that's one case we know some about.
And Eileen Jaffe points out there's an argument that the EGF receptor functions as a tetramer of dimers and along the same kind of thinking. Well, I'll switch to a little bit different question. Carvalho de Sousa asks, question about direct coupling versus swapping domains. It's known that a single amino acid changes can change the folding rate of the channel. What have you heard being 30 plus years in the field, what have you learned regarding rigor when before nailing an ion channel? I'm not quite sure, but I think the question really has to do with the role of domain swapping and whether that's something that evolution is using, or it's something that just comes up artificially in the experiments.
Okay, so I'm not sure exactly what the question is getting at, but I'll take an attempt. There have been some proposals that maybe a single protein can adopt both conformations. And there is one case with a particular trip ion channel, and I think that's what the questioner is asking about, where one channel exhibited a domain swapped and then in a not domain swapped case, and it was a mutant versus non. And so the question is, does that particular channel do both, or can this be a mistake? Well, we've looked at, on that dendrogram, actually, if you look at it in the related channels, and we know many of the structures now, you can see that actually nature made ones that sort of are all most closely related that are domain swapped, and made some others that are not. And we have not seen any cases with the potassium channels we've studied where you see one that can do both. So that's our experimental observation so far. And based on that, I would say we have two kinds, and for the channels I've studied, they don't swap in between them, or at least we have no evidence for that. But one always keeps an open mind as an experimental scientist. Yeah.
So there's one more question I'll cover, which is in calcium and voltage activated potassium channels, even with the right combination of depolarized membrane potentials, at the single channel level, the open probability tends to be high, but however, there's always closed states that are observed. It's never fully open. So what is the mechanism that explains this?
Yeah, so there are channels that actually, when they're fully activated by ligand or voltage, that may achieve an open probability of 5% maximum. And there are other kinds that can achieve almost 100%. And what happens is there's a voltage sensing mechanism or a ligand-gated mechanism, but the ion conduction pathway can get occluded elsewhere. For example, even the selectivity filter may fluctuate, or elsewhere along the pathway you can get a closure. So imagine you could open a main gate, but once that gate is open, you don't get to see a fully open, always conducting channel. You actually have another part of the channel that is fluctuating and occluding the pore. And really, really opening the main gate is just unveiling that process. That's probably similar to what you saw in the channel recordings in an earlier talk by Ju Chen with the CFTR, when it could be activated, yet you still saw the channel fluctuate. In fact, it would be the exception for a channel with sort of a single regulated gate that is open to 100%. Although I have to say, we have channels that go open to 99% and then you could ask, well, I think a lot of the way we study these channels in the biophysics lab, we like to drive, we like to add our ligand or depolarize our membrane to we cover the whole range of the activation curve. But if you think about it in nature, and you think about the capacitance of a typical cell, and the conductance of a channel, and then you realize just a few of those channels have to be open for a few milliseconds in order to completely polarize or depolarize the membrane, you realize that probably nature isn't usually operating over that range that we study in the biophysics lab. In fact, it's just a few flickers open, and the channel has already achieved its purpose in a sense. It's fulfilled its function. That would be my answer to the question.
I think nonetheless, even the open state is multi-conformational states, as you say, because it's sort of multi-gated.
Yep.
And there's probably some evolutionary pressure to do that, in fact.
Part of it may be an evolutionary pressure, and part of it may be just proteins fluctuate.
Mm-hmm. Yeah. Okay, Rod, thank you very much. It's great to hear that perspective and also the new thing. So thank you very much. Thanks for joining us.
Thank you.